A neuron produces an output that's carried to perhaps thousands of target neurons. Each of those target neurons could also get inputs from thousands of other neurons. And from the current state of all these inputs, that target neuron must determine whether or not to produce its own output. So, how is all this information transmitted between neurons and received in such a way that it can be appropriately integrated? Well, the primary way information is represented so that it can be transmitted and integrated is by electrical signals called action potentials. So first, let's have a closer look at the action potential that propagates along an axon. It's a spike of voltage, about 100 millivolts, lasting about a millisecond or two. Action potentials actually propagate along the membrane with relatively little disturbance to the interior of the axon, and action potentials along an axon are all or nothing. To a first approximation, you don't get smaller or larger voltages, longer or shorter durations, and the same signal is received by all the synapses onto all the different target neurons. Now, action potentials of various kinds can also be generated within the dendrites, again propagating along the membrane or cell wall. But in this case, the action potentials can gradually decay, although the decay is slower than would occur as a result of simple electrical resistance. In other words, there's partial signal regeneration. Also, action potentials initiated at different points in the dendrite can meet, for example at the junction between two branches, and reinforce each other, so that a somewhat larger combination signal propagates deeper towards the soma, but still decaying slowly. Looking closer at a branch on the dendrite of a cortical pyramidal neuron, all the excitatory inputs from other pyramidal neurons are actually located on the tips of their own individual spines that stick out from the trunk of the dendrite. Spines are significant because they allow the chemistry at one synapse to take place independent of the chemistry at other synapses. But sometimes there's other synapses from different types of neuron close to a spine or on the branch. An output from a neuron into its axon is this voltage spike. The spike travels along the axon until it reaches a synapse on the dendrite of a target neuron. When the action potential arrives at the synapse, it triggers a reaction. That reaction is injection of a postsynaptic electrical potential into the dendrite. In the case of excitatory synapses on the tips of spines, this potential is electrically positive and encourages the target neuron to produce its own output action potential. The injected potential resulting from one spike at an excitatory synapse increases rapidly after the arrival of the spike and then decays more slowly over a period of perhaps 8 milliseconds. Now, although action potential spikes are almost identical, synapses differ, and they differ in the amount of potential injected into their target dendrite in response to one action potential. In other words, for the same action potential, they differ in the degree of encouragement they give to their target neuron. Let's suppose that over a period of time, a number of action potentials arrive at a group of synapses on the same branch, and I've illustrated them by the blue spikes. Each spike injects positive postsynaptic potential into the branch, varying depending on the strength of the synapse. The postsynaptic potentials can add together to create a total branch postsynaptic potential. And if the total at some point exceeds a threshold, a dendritic action potential will be launched from the branch and travel deeper into the dendrite towards the soma. But remember that each postsynaptic potential decays over a period of about 8 milliseconds. So if two action potentials arrive at nearby synapses on the same branch, but are separated in time by more than 8 milliseconds, the first potential will have decayed by the time the second begins, and they won't reinforce each other. So you have to have enough action potentials arriving within a fairly short period of time to get to that threshold. In other words, you can effectively define an integration window in time. If enough spikes arrive on the same branch, the dendritic branch will produce an output, provided the spikes arrive within one integration window. Now, some synapses may be inhibitory or inject negative potential that opposes the production of an output action potential by their targets. And in the case of the inhibitory potential, it reduces the total potential, reducing the chance that the threshold for an output will be reached. But again, there's a period of time over which the inhibition operates. So let's have a look at what's going on at steadily increasing scales. Action potentials arrive at synapses, and synapses are grouped on branches of a dendritic tree. Branches are linked together. Multiple groups form a dendrite, and multiple dendrites terminate on the neuron soma. So if within the integration time, enough action potentials arrive at different synapses on the same terminal branch, and I've indicated the arrival of an action potential by colouring the synapse on its spine green, if there's enough, then a dendritic action potential will be initiated out of the branch, travelling deeper into the dendrite. Depending on the number and relative timing of the incoming action potentials, the size of the dendritic action potentials can vary. Dendritic action potentials, as I mentioned earlier, they decay as they propagate, and the distance one potential reaches is limited. However, at a junction between two branches, two dendritic action potentials can reinforce each other, leading to a larger combined potential that can travel further, where one action potential alone would decay away to zero. Although a dendritic action potential from just one branch might propagate further if it was very large. At the dendrite level, the dendritic spikes generated in terminal branches are integrated. For example, two spikes from adjacent branches may reinforce, join with a large spike originating in another branch that decays but still has some amplitude when it reaches a junction with the other branches, resulting in a dendritic spike which propagates on reaching the soma and perhaps triggering an output action potential from the neuron. Any dendrite could contribute. Perhaps if one dendrite doesn't have a combination of inputs sufficient to reach the soma, other dendrites, or perhaps even a combination of dendrites, could trigger an output action potential. So the synaptic inputs are integrated by a very complex algorithm to determine neuron outputs. And of course, inhibitive inputs affect the integration process, reducing the effect of an individual branch, or of an individual synapse, or of an individual dendrite, or even blocking the neuron as a whole from producing an output regardless of the state of its inputs.